Hello, in today's video, we shall learn in detail about transpiration in plants. By the end of this video, you will have a clear understanding about different types of transpiration, structure of stomata, external and internal factors affecting rate of transpiration, ascent of sap, opening and closing of stomata, active potassium theory, advantages and disadvantages of transpiration. Let us start with transpiration and its types. Loss of water in the form of water vapor from the aerial parts of the plant is known as transpiration. The different types of transpiration are lenticular transpiration, cuticular transpiration, and stomatal transpiration. In lenticular transpiration, loss of water from plants as vapor occurs through the lenticels. Lenticels are tiny openings present on the stem of many woody plants. A lenticel is a porous tissue with large intercellular spaces in the periderm. These pores in the tissue act as a pathway for gaseous exchange and transpiration. Water vapor loss is 0.1%, which is negligible as compared to total loss. In cuticular transpiration, loss of water from plants as vapor occurs through the cuticle. Cuticle is a layer of wax-like coating on the epidermis of leaves and stem. Transpiration is by direct evaporation from outer walls of epidermal cells through cuticle. Rate of evaporation depends upon the thickness of the cuticle. That is, greater the thickness of the cuticle, lesser is the evaporation. Xerophytic plants generally have a thick cuticle and a waxy coating on the leaves and stems to check cuticular transpiration. Water vapor loss is 3 to 10% of total loss. In stomatal transpiration, evaporation of water takes place through specialized pores called stomata, which are present in the leaves. Water vapor loss in this case is 90 to 97% of the total loss. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Let us look into the structure of stomata. Stomata are minute pores present in the epidermis of leaves. They help in gaseous exchange during respiration and photosynthesis. Minute pore stoma is surrounded by a pair of guard cells which are kidney-shaped in dicots and dumbbell-shaped in monocots. Each guard cell has a dense cytoplasm with a nucleus and number of chloroplasts for photosynthesis. Subsidiary or accessory cells surround the guard cells. Next, we shall learn about the factors affecting rate of transpiration. The rate of transpiration in plants depends on various external factors and internal factors. External factors are temperature, relative humidity, wind speed, light intensity, etc. Internal factors are number and distribution of stomata, orientation of the leaf, root-shoot ratio, leaf area, leaf structure, etc. Let us look into the external factors. First is temperature. Rate of transpiration is directly proportional to the temperature. High temperature decreases relative humidity outside the leaf. There is increase in water potential gradient, due to which rate of evaporation increases and hastens the loss of water. Relative humidity. The relative humidity of the atmosphere is inversely proportional to the rate of transpiration. If leaf is surrounded by moist air, due to increase in humidity, rate of diffusion of water vapor decreases. When the atmosphere is dry, humidity is less and there is increase in rate of transpiration. Wind speed. Still air lowers the rate of transpiration. Water vapor accumulates around the transpiring organs, causing lowering of rate of diffusion. The movement of air speeds up diffusion of water molecules into the surrounding air and increases the rate of transpiration by removing the soil air around the leaves. Light intensity. In majority of plants, stomata open in the presence of light and close in darkness. So, rate of transpiration is directly proportional to light intensity. The opening of stomatal pores helps carbon dioxide to enter the leaf and thus supports photosynthesis. Atmospheric pressure. Rate of transpiration is inversely proportional to the atmospheric pressure. Low atmospheric pressure enhances evaporation, produces air currents, and increases the rate of transpiration. Availability of water. Rate of transpiration is directly proportional to the amount of water present in the soil. When there is a decrease in water uptake by the roots, the leaf cells get dehydrated, resulting in the closure of stomata and wilting of the plant. If rate of transpiration is more than the rate of absorption, the stomata close and the plant wilts. Wilting is defined as the drooping, folding, or rolling of leaves and young stems due to loss of turgidity. It occurs due to high rate of transpiration and low rate of water absorption. The different types of wilting are incipient wilting, temporary wilting, and permanent wilting. Incipient wilting. In incipient wilting, there is partial loss of turgidity in the plant cell, but the symptoms are invisible. Temporary wilting. During hot summer days, temporary drooping of leaves and shoots occur due to high transpiration rate causing water deficiency or loss of turgidity. When the temperature decreases at night, the plant regains water and normal turgidity. Permanent wilting. When the plant cells do not get water from any source, the absorption of water ceases and the cell pass into a state of permanent wilting. These cells do not retain their normal state even after maintaining a suitable environmental condition. Altitude. At high altitude, the atmospheric pressure is low, resulting in more transpiration. In order to reduce rate of transpiration at high altitude, the plants develop certain xerophytic adaptations. Now, we shall learn about the internal factors affecting rate of transpiration. Leaf area. Bigger leaves or leaves with larger surface area show greater water loss due to transpiration. Structure of leaf. Thick cuticle, sunken stomata, reduced size of leaves, compact mesophyll cells, presence of multilayered epidermis, epidermal hairs on leaf surface cause decrease in rate of transpiration. Number of stomata. Rate of transpiration increases with increased stomatal frequency. Mucilage and solute. Presence of mucilage and solute in succulent part of plant reduces rate of transpiration. Modifications. To reduce rate of transpiration, leaves are modified into spines or phyllodes, and stems are modified into thorn, prickle, phylloclade or cladode to reduce rate of transpiration. Root-shoot ratio. Low root-shoot ratio decreases the rate of transpiration and higher ratio increases the rate of transpiration. An extensively developed root system is more efficient in water uptake from the soil. Increased availability of water increases rate of transpiration. Now, let us understand ascent of sap. The upward translocation of water along with dissolved minerals from the roots to the aerial parts of the plant is called the ascent of sap, which takes place through the xylem vessels. The water with dissolved minerals is called sap. Various theories were put forward, but the most accepted theory is transpiration pull or cohesion tension theory. This theory was proposed by Dickson and Joly in the year 1894. Main features of this theory are strong cohesive force of water, continuity of water column in the plant, and ascent of sap due to transpiration pull. Strong cohesive force of water. In the water column, there is strong attraction or cohesion between the water molecules, due to which they cannot be separated from each other easily. There is also attraction between water molecules and the inner walls of tracheids and vessels of xylem, which is known as adhesion. We all know that the walls of xylem ducts are made up of lignin and cellulose, which shows affinity for water. Continuity of water column. It is maintained by the cohesive and adhesive properties of water. The water molecules adhere to the walls of xylem due to its adhesive property, and they remain together and move up as a continuous water column due to its cohesive property. Transpiration pull. During transpiration, water molecules evaporate from the surface of leaves through stomata. The concentration of water gets reduced in mesophyll cells, and diffusion pressure deficit of water in mesophyll cells increases. A negative pressure in xylem vessels pulls the water from the soil. The surface tension of water and capillary action facilitates the movement of water through the plant. Transpiration and guttation are the two physiological events in plants by which they release water to the atmosphere. Next, we shall learn about the mechanism of opening and closing of stomata. Various theories have been put forward to explain the mechanism of opening and closing of stomata. God's cell photosynthesis hypothesis. The opening and closing of stomata is controlled by God's cells. During the day, due to endosmosis, the stomata becomes turgid. The turgor pressure stretches the outer thin walls of God's cells outwards. The inner thick walls are pulled apart, resulting in the opening of stomata. When God's cells lose water by exosmosis, turgidity decreases and the cell becomes flaccid, causing stomatal closure. During daytime, in the presence of light, the guard cells of the stomata photosynthesize with the help of chloroplast, forming sugar. The soluble sugar increases the osmotic concentration of the sap of guard cells. Endosmosis of water takes place from the neighboring cells and the guard cells become turgid. This results in the opening of stomata. At night or in the absence of light, there is no photosynthesis and the sugar present in the guard cells gets converted into starch. Since starch is insoluble, the osmotic concentration of guard cell decrease. Exosmosis of water takes place from the guard cells and the guard cells become flaccid. This results in the closing of stomata. A high pH favors opening and a low pH favors the closure of stomata. In the presence of light during the day, carbon dioxide concentration of guard cells is lowered due to photosynthesis and hence there is rise in pH. This favors conversion of starch into sugar, which increases the turgor pressure in the guard cells and the stomata open. In the dark, due to respiration, carbon dioxide accumulates in the guard cells, resulting in the fall in pH of the guard cells. This favors conversion of sugar into starch, which decreases the turgor pressure in the guard cells and the stomata close. During transpiration, as water evaporates through the stomata, there is a continuous pull of water molecules into the leaf from the xylem. The force generated by transpiration creates sufficient pressure which helps to lift water over several meters high. Now, let us understand the active potassium theory. According to Leavitt 1974, Opening of stomata is due to the entry of potassium ions into guard cells from surrounding subsidiary cells and epidermal cells, since it increases the osmotic concentration of guard cells. Uptake of potassium ions requires energy in the form of ATP, and it also involves hydrogen and potassium ions exchange. During daytime, starch present in the guard cells is converted into phosphoglyceric acid and then to malic acid in the presence of enzyme phosphenol pyruvic carboxylase. Malic acid dissociates into hydrogen ions and malate ions. Hydrogen ions from the guard cells move to the epidermal cells, and potassium ions from epidermal cells are absorbed into the guard cells with the help of ATP molecule. Increased potassium ions in guard cells increases their osmotic concentration. Endosmosis occurs and water enters the guard cells. This increases the turgor pressure of guard cells, leading to the opening of stomata. During night time, in the absence of photosynthesis, the carbon dioxide concentration increases in the substomatal cavity. Inhibitor enzyme abscisic acid inhibits the uptake of potassium ions. Potassium ions are transported from guard cells to subsidiary cells and epidermal cells. This results in decrease in osmotic concentration of guard cells. Exosmosis takes place and water moves out from guard cells, making the cells flaccid. Let us understand the term antitransparence. Any substance or material that reduces the rate of transpiration is called antitransparent. They are of two types: metabolic inhibitors and surface films. Metabolic inhibitors, without affecting other metabolic activities, reduce the stomatal opening for a short period of time. Example: phenyl mercuric acetate, acetyl salicylic acid, abscisic acid, etc. Film forming chemicals check transpiration by forming a thin film over the leaf to cover the stomata. Example: dimethylsilicone, polyethylene, plastic films, etc. Sometimes wax emulsions are sprayed as film coating compounds to cut down transpiration. There are several advantages of transpiration. Ascent of sap. Transpiration helps in the transportation of minerals which are absorbed by the roots from the soil by creating a transpiration pull. It helps in the distribution of minerals. Transport of water. Transpiration helps in transporting water to the entire plant by creating a transpiration pull and maintaining a continuous water supply through tracheids and vessels of xylem. Cooling effect. Transpiration gives a cooling effect to the plant by lowering down the temperature through evaporation of water. Maintains turbidity. Transpiration maintains the shape and structure of plant parts by keeping the cells turgid. Helps in photosynthesis. transpiration supplies water for photosynthesis and is also responsible for the opening of stomata, which lets in carbon dioxide from the atmosphere inside the leaf for photosynthesis. Finally, we shall look into the disadvantages of transpiration. Wilting. During noon, rate of transpiration is higher than the rate of absorption. This results in loss of turgidity or wilting. Wilting reduces photosynthesis and other metabolic activities. Reduced growth. Transpiration results in water deficit within the plant cell, resulting in decrease in growth. Reduced yield. Decrease in availability of water inside the plant affects the meristematic activities and affects the formation of flowers and fruits. Abscisic acid. It is a growth inhibiting hormone produced within the plant during water crisis. This prevents various plant activities and promotes abscission of leaf, flowers and fruits. Wastage of energy. Since a large amount of water is lost through transpiration, the energy used by the plants for absorption and conduction of water from the roots to the aerial parts goes waste. So today we have learnt in detail about transpiration, various factors affecting transpiration, its advantages and disadvantages, and also the various theories related to the mechanism of opening and closing of stomata. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.